Thank you, everybody, for coming out to the 30th edition of the Multiomics Reading Group. I'm your host for today, Cesar Miguel de Córdoba. And today we have the absolute honor and pleasure to have Xun Xu presenting his amazing work featured in the science journal called Stereo-seq and Stereo-cell Technology Progress. Or, well, that's not the title of the journal, that's the title of the talk. I'm sorry about that. But we're going to have him presenting about this general research direction. I would like to make a note and say that today we have the particularity of celebrating our 30th edition of the reading group. So we will be hosting from both Mila and the Alberta Machine Intelligence Institute, where I happen to be situated in this day and time. So without any further ado, having these many institutions and people in the call, please, Professor, take it away.
Yeah, so
okay.
I need to give some introduction for our honored guest here.
Sure, my bad. I completely overlooked that. Can you please do your wonderful introduction as you usually do?
Yeah, thanks. Okay, thank you, Professor Xun, for coming to our reading group. So, hello everyone. Professor Xun is actually the chief scientist from BGI Research and also the director of State Key Library of Genome and Multiomics Technology. So he's also a PhD supervisor of UCAS and also Wuhan University. He serves as the convener of WG6 for biotechnology community and also member of SACTC 559. So his research mainly focuses about developing sequencing, single cell, and also stereomics technology. He designed and developed the first BGI sequencer, as well as the DNB Lab C4, stereo-seq, and also the stereo cell technology. He also made his effort in applying all this technology in understanding biodiversity and applications in agriculture and also disease research. Professor Xu has published more than 200 scientific peer-reviewed papers with more than 17,000 of citations in last 5 years, and selected 9 times as the Clarivet highly cited scientist. So it's very honored to have you here, Professor Xun. So please have it. Thank you for coming.
Okay, thank you. Thanks a lot for the nice introduction. So as I mentioned, in this first page is actually our new headquarters. It's actually located very close to the very nice beach, and also the Orix Island is island from Hong Kong. So it's really nice. And today, my topic will mainly focus on the stereo cell. But before I introduce more on the stereo cell, because stereo cell is based on stereo-seq, so I will introduce a little bit about the stereo-seq technology. So I was actually, when I joined BGI, I was started to working on the genome sequencing, but the genome itself cannot help us to understand all this mechanism behind the biological process. So that's why, besides the genome sequencing, we also have to understand those a complex biological process by single cell level. So that's why we do a lot of single cell sequencing. And in the recent 5 years, single cell cannot fully help to explain all these cell-cell interactions and the cell dynamics. So that's why we have to understand the cell in the spatial and temporal level. That's why we call it stereomics. So in this cell review paper, I try to explain why these 3 parts, including DNA sequencing, cell omics, and stereomics, are very important for us to understand the process and the mechanisms behind all these biological phenomena. So as you know that compared to the bulk omics and the single cell omics, spatial omics provide the more detailed information, not only the expression level, but also the locations and the cell-cell interactions. So that's why, compared to the sequencing and the single-cell omics, spatial omics give us a lot of opportunities to understand those complex process of biological features. So, but as you know, there's a lot of limitations in the previous technology. Like one is the capture efficiency, and the other one, the limitation is the resolutions. So our goal is trying to develop a technology that have a very high gene capture efficiency and also have a very high resolutions that can help us to do the single cell segmentations. So that's why we developed our technology. We call it StereoSeq technology. So this StereoSeq technology is based on our sequencing platforms. So our sequencing platforms is actually based on the silicon wafers. So in the silicon wafer, we have modified the surface of the chips with a patterned array. What do we call pattern array? The pattern is kind of like a 200 nanometers size as a small dot. This small dot is kind of able to attach the DNA nanoballs on this small dot. So, and the distance between two spots is around like 500 nanometers. So as you can see that this is actually very high resolution compared to the other spatial omics technology, because they are using the printing technology. The printing technology, they can only able to reach to like a micro level compared to our like 200 nanometers resolutions. So that's why we have a very high resolution, probably the highest of all these like commercial available spatial transcript technology. And because we are using silicon wafers, so the largest one we could have 13 centimeters by 13 centimeters. Then we're using the laser to cut into the small pieces. So which means we get very flexible on the size of the chips. We can have like 1 centimeter chips, but also can have a 13 centimeter chips. So then we label these DNA nanoballs with random sequence, like each 2 spots in these chips. This probably we have probably more than like 10 or 100 billions of different spots on these chips. Like each spot has a very unique sequence, and then we do first round of sequencing, and we are able to know this like physical location of each unique random sequence. So we're using this unique sequence as a barcode for those locations. And then we ligate this DNA nanoballs with poly-T. Poly-T tail, we're able to capture the RNA fragments. So if we put the fresh frozen samples on the chips, these chips can capture the cDNA bodies, but also we will, when we do the reverse transcripts, we'll have also the cDNA sequence as well as the location barcodes. After the second round of sequencing, we were able to know where like each of these cDNA, the physical location, are really located. Then bioinformatically, we convert back to the real tissues. So by this way, we are able to have a spatial resolved transcript profilings. So because the high resolutions, and we also can combine with the imaging data, for example, including like nuclear staining or the cell membrane stainings. Using this information, we can do single cell segmentations. So single cell segmentations is very important for us to understand cell niche, because as you can see, if without any single cell segmentations like using the Bin50s, you are missing all these like cell-cell interactions and the cell niche informations. But if we can do single cell segmentations, you can see like in the very small regions, how these different type of cells are interact with each other. So that's why the single cell segmentation is a very important steps, and it's very important features of our technology that we can do single cell level spatial transcripts. And by this technology, we are using this for understand like the brain. The axolotl brain regeneration projects we published in Science in 2022 is the first paper we are able to do the single cell segmentations. And later in the 2023, we're able to build up the 3D macaque whole brain. And into 2024, last year, we are able to do the 3D for the mouse embryos. So, but this technology is very limited on fresh frozen samples. But like in the clinical fields, most of the samples are FFP samples. And so, and the current technology are also limited by the 3' end of RNAs and cannot capture the 5' of RNA and cannot do the alternative splicing. And it's also can only capture the messenger RNA because we using the poly-A tail as a capture probe. So we want to expand it from messenger RNA to the full all the different types of RNA. And also the requirement for samples, for example, FFP samples cannot use before. So because of that, we recently able to solve this version 2 technology. So the version 2 technology is using the random probe to capture the RNA instead of using the poly-A tail. So which means even this highly fragmented RNA without poly-A tail, we can still capture them. And we also able to do the in-cell RNA cDNA synthesis instead of like released RNA from fixed samples. So which means like for FFP samples, we are much easier to do the to release RNA from the samples. So because of that, in this the second version of the technology, we can do whole transcript and able to do the full length of RNA. So as you can see that we can have a very even distribution across the whole gene body, including 3' and 5' of RNA. While the other technology, including our version 1 technology, can only capture the 3' of RNA. Because we can do the full transcripts, we can do the alternative splicing analysis. And because we are not only capture poly-A tail, so that's why we expanded from the messenger RNA to all these like the non-coding RNA, especially for those long non-coding RNA. And it's also showing a very robust performance on clinical FFP samples, including like those samples has been for more than 8 years. We can still have a very good capture efficiency. And another very important like features by this technology, we can co-detect the host and the microbiome on the spatial level. So because in the microbiome transcripts, because they won't have a poly-A tail, so the previous technology cannot capture that because we are now using the random probe. So that's why we can capture both the microbiome and the host at the same time. For example, this is a TB infection. You know, in the human TB infection model, the bacterial level is really low. So our technology is very sensitive that we can capture those TB bacteria. And at the same time, we can also capture the human cells, the human, like for example, B cells, because we can capture the both the 5' and the 3' RNA. So the 5' RNA we can use for this like BCR clone, capture and assembly. And those BCR clones, these antibodies, then later we can also detect in the human blood. So which means this is a very sensitive and accurate technology for this like host and a microbiome infection model study. So as a summary, that our like version 2 technology is able to solve those like clinical FFP samples and have capabilities with RNA degradations as well as the host and microbiome transcripts. So after that, because this can do the single cell spatial omics, then later I came back to ask a question: can this technology be applied for the single cell omics? For single cell omics, as you can see that in the past 20 years, there's almost like more than 20 years, there's a lot of technology has been developed to be able to enrich like a high throughput and a multi-omics information in a single cell level. But actually I studied on I start on working on the single cell technology in 2009. At that time, we are using the like a very low throughput technology, using the mouse peptide to pick up each cells and then amplify its genome and sequence. So it's very low throughput technology. Later, we try to develop much higher throughput technology, for example, using those like microwell to capture the cells. And later, we're using the fluidic-based technology to capture the single cells. But still, all this technology still have a lot of bottlenecks. So one of the bottleneck is the throughput, the flexible throughput, because those like the capture efficiency is not that high. Like the only most likely, for example, in the fluidic-based technology, we can only capture like 40 to 60% of cells in a level of like 10,000 cells. If the input cell is less than 1,000 cells, for example, 300 cells, the current fluidic-based technology cannot capture those low, small starting materials. If we want to do like, for example, million cells, they have to divide it into different batches. So it's not very flexible in the throughput. And when we try to using for different purpose of studies, sometimes you cannot starting from very small throughput or ultra high throughput. So the second problem for the current technology is that it can only capture like a standard round shape small cells. If the cell is like a larger cells, for example, the muscle cells or neuron cells, the current technology cannot capture the single cell level. Most likely they will just digest into the nucleus. They then do single nucleus capture. And sometimes because they have like a strong bias, selection bias on the round standard cells shape. So that's why they have a very strong bias on the size of the cells. If the very small cells or very large cells, most likely will have less preference by the current fluidics or droplet-based technology. The third limitation is that the current technology can only capture the molecular information, the RNA information, but we never know what the cell really looks like because they cannot do the imaging data, cannot generate the imaging data. So, but the imaging data is really important for us to know which cell are they and what they looks like. And currently all these like the single cell technology are using like cell distortions. So which means all these cells are released from their original tissues or original environment. So they will lose a lot of like informations. So our technology is based on the stereo-seq technology, as I introduced previously. So the way is that we pre-coding the chips with a kind of like a glue we call poly-L license, which is able to attach the cells on the surface. And then we directly dropped the cell suspensions on the chips. And in these chips, this like a poly-L license will capture all these cells in the surface. And after that steps, we will do biochemistry and staining using different staining methods, using different antibodies to generate the imaging information. So we are able to see what are those cells looks like and what their like the surface markers looks like. And after that imaging steps, then we captured RNA and sequence them as the same protocol as I introduced before using the stereo-seq. So as you can see that because we can then later convert back to the imaging data. So it's directly combination the imaging data as well as molecular information. So this is how it looks like in the imaging data. So we mix the human and mouse cell lines on this 1 by 1 centimeter chips. So because the mouse cell lines and human cell lines, they have their own surface markers. So we can see how these two type of cells mixed together, how they looks like in the same chips. So as you can see that because we have the imaging data before, like in single cell sequencing methods, there's one very important bottleneck is kind of like what we call the doublet, which means there's one data point mixed by two cells. So they are not real single cell level. Because we have the imaging data, so it's really very simple way that you can able to see which data point mixture of two type of cells. So using that way, you can remove all these like informations. So this is a easier way that in our methods that we can able to reduce the kind of like the contaminations of what we call the doublet. So this is how the data looks like. Before we move those like doublets, we can still have like more than 200 like spots actually mixed by human cell line and the mouse cell line. But after using the imaging data, we can remove most of those like doublets. From the sequencing results, we can see that this doublets is much lower compared to before. So, and this is the way that we can significantly reduce like contaminations by mixture of two cells or multiple cells instead of like a single cells. So, and we also have the comparable capture efficiency compare all these commercial platforms. So this is the results from the PBMC. As you can see that we have a slightly higher gene capture efficiency compared to the commercial platforms. And another very important features, as I introduced earlier, is that we have less bias, capture bias compared to the other fluidic or droplet-based technology. As you in the PBMC cells, because they have different shape of cells. The monocytes, because they are like a small round standard shape of cells, so they are more preferred to be captured by droplet-based technology. So that's why their percentage is much higher compared to the fluidic-based analysis. But in the real world, that this monocyte cell type is around like 18%. So our results showing that we have the same similar like the percentage of different type of cells compared to the flow cytometry analysis, we have much less bias compared to the fluidic or droplet-based technology. And we have a very flexible cell input range from 200 to 20,000 cells. For example, in this study, we input for 200 cells. In this experiment, we input 2,000 cells. So which means for those like a very small amount starting materials, like sometimes people just select cells from the flow cytometries. The target cell, the number is only like a few thousands or only few hundred. At that time, it's really difficult to use in the current like fluidic-based technology. So our platform's showing that it's much easier way in this platform to do like the small starting materials. And because we can using different size of the chips, so we have a very flexible throughput, like in the S1, which means it's 1 centimeters by 1 centimeters chips. We can input like 25,000 cells. In this like 6 by 6 centimeters chips, we can input like 1 million cells. If we're using our largest cell, the chips, like 13 by 13 centimeters chips, we can input like 10 million cells. So as you can see, that's very flexible on the size of the numbers of the cells. So that's what's the application for those like a very large amount of cells, because sometimes we want to find out all these like very rare cell types. So for example, in PBMC, there's a particular like a very rare types of cells, which is HSPC. So it's kind of like a stem and the progenitor cells. So it's the percentage is very low. It's kind of like less than 1 in 1,000. So in this like a very high throughput chips, like 3 by 3 centimeter chips, we're able to capture them, and it's much more obviously in like a 6 by 6 centimeter chips. So we can capture more than like 100 of them. So this is very important using like a large chips to detect those like very rare cell types. And because, as I mentioned, our technologies are compatible with imaging like kind of staining protocols. So we can using like multiple immunofluorescent staining to label the cell surface markers. So in this data set, we're showing we can using 3 different cell surface marker to staining the immune cells. And this immune staining imaging data can also combine with the sequencing data. And furthermore, we can expand it from only like several like staining steps to the whole protein analysis. So the way is that we're using a protocol we call the site-seq. So we labeled the antibody with DNA barcodes. So then we're using this like labeled antibody to able to staining the chips on the cells on the chips. So because we can have like more than 300 different types of antibodies to staining at the same time, and we can sequencing the protein barcodes and the RNA at the same time. So using that way, we can have like more than hundreds of different antibody targets together with the transcript data. So this is the protocol we call the stereo cell site-seq. So this actually gives us much more information, not only RNA, but also the protein. So the benefits by adding the protein information is because sometimes the RNA expression level is really low, especially in this like immune cells. So, but in the protein level, those genes are much higher abundance. So using that way, we can able to identify those very unique immune cells. For example, in our study in the human PBMC, we're able to identify a new cell type. So this new type of cell type in human PBMC is actually, if you only using the RNA sequencing data, you cannot identify them. But if you add in protein information, you are able to identify these small proportions of cell types. These novel cell subtypes is may associated with T lymphocyte tissue migrations. So this is then later we are able to verify this new cell, this new novel cell types. And in our study, we are, hello?
Yes, can you go back on the slide? Back up a slide, please.
Yes.
Yeah. So can you confirm that once you have found this yellow cluster additional from the site-seq data, was there a difference in the RNA expression between this yellow cluster and this neighbor blue cluster, or was it purely on the site features that there was a difference?
In the RNA expression levels, you cannot tell the difference between these blue portions and the yellow portions. But if you do the detailed analysis between these two portions after you able to differentiate them, you can still find gene expression differences.
Okay.
But only in the clustering informations, they actually cannot tell the difference between them.
Okay.
But they do have gene expression differences.
But like, I guess if you had taken this GDT cluster, only this cluster, and tried to subcluster it, don't you think you would have found this CD112 plus cluster? I guess my question
Yeah.
is not fair because it's not what you do. But,
Yeah, but if you do that, I think you can still see they actually have some differences.
Okay.
But they are not that significant. Most likely, you are not able to identify them as like a subtype. Yeah.
Okay. No, I agree.
Yeah, actually, because there's a lot of like a PBMC study before, but none of them have been reported this small part. Yeah, it's because
Okay, and how much features did you use for the site-seq experiment compared with how many gene variables you used for this UMAP embedding?
So for the for genes, I think we using all these genes we detected. For the protein, we have in this study we have 154 targets.
So it's like 100 versus 20,000.
Yes,
but still these 100 are significant. Okay, great, thank you.
Yeah, and one more thing is that for this like CD112. This kind of like proteins in their expression level, because if their expression level is really low, it's really difficult to detect them in the transcript level in this portion of cells, but they actually very high level in the protein level.
Okay, amazing, thank you.
Yeah. And another like features of our technology is that we can do like on-chip cell culture, and we then do in situ detection. So we found our chips is very robust in the cell culture systems. So we show you that our chips can stay in this culture dish for more than 2 months, and after 2 months, our chips can still play the functions to capture the transcripts. So, which means our chips can be used in the cell culture protocols. So then we did cell cultures using 2 different conditions. One is like normal conditions. The other one is LPS conditions. So then we try to see in these 2 culture systems whether or not we can see they are different in these 2 different conditions. As we can see that they actually showing a very interesting difference between the control and the LPS conditions, because a certain kind of like they actually have a small subgroups in this like 0, 6, and 9. They are actually not real cells. They actually are the extracellular vesicles. So because although they are very small, but we can still capture the RNA from this extracellular vesicles. So, and they're also showing a little bit difference between the control and the stimulations. And the interesting thing is that in this subcluster 4 and 3 actually are very different between the control and the LPS stimulations. LPS stimulations, we're getting much higher number of subgroup 4 and subgroup 3. So, but if you without like this kind of like in situ detections, if you just using like cell suspensions, they're like separate filter all these cells, separate these cells, and using the standard single cell sequencing protocols, you won't see these differences, and you won't see all those like extracellular vesicles. So this is showing you that comparing with our own chip cell culture and dissociated samples. So if you without this on chip cultures, you will see that you cannot see these differences. As you can see, this in situ captures, they actually have changed a little bit. But if you using the chips or using the chip or using this dissociate cell samples, they're actually showing more or less the same. But you cannot see this blue subpopulations. So which means in situ captures maintain a high fidelity transcriptome. The transcriptome is actually changed when you propel the cultures. So that's why we believe that in situ detections can be very sensitive for a lot of small changes during the cell culture. So, and we further trying to see this a co-culture, can we detect a lot of more information using two type of cells co-cultures. So this slide is not published in our paper. We just recently working on it. That we're using the cancer cell lines and immune cells to co-culture together to see if those immune cells can recognize certain kind of tumor cells. So in this preliminary very early stage results, we're showing that this kind of co-culture can able to see cell-cell interactions between the tumor cells and the macrophage cells on our chip surface. And another important thing compare our platforms with other fluidic-based technologies that we can really able to working on the true cells, which means we can do those muscle fiber cells or neuron cells. So before, when we working on the muscle cells or the neuron cells, we were only able to working on the nucleus instead of the whole cells. So, but the whole cells and the nucleus are very different to each other, especially for those muscle cells. So in the muscle cells, like the muscle fiber cells, they are very strange cell type that they are very long, very large. They have more than hundreds of nucleus in one cells. So the question is whether or not those different nucleus in the same cells express the same genes. So our results showing that in different nucleus in the same muscle cells, they actually could express different genes. So for example, here in this particular type of muscle cells, EDL, in different parts, they express different group of genes. For example, in the neuron muscle junctions, and kind of like a neuron, the muscle cell junctions, they actually express different gene patterns. And we are also trying to apply this on another very large cells, the oocytes. The oocytes, because the size is really large, that sometimes it's really difficult to using the standard single cell technology because the fluidic or droplet-based technology cannot capture them. So the only technology can do is what they call smart-seq, but it's a very low throughput in using the single tube to do the single cell expression and sequencing. So it's very low throughput technology. So, but our platforms, you can directly drop the oocytes on the surface of the chips, and you can in one single experiment you can have a few thousand oocyte cells at just in one time. So here in our experiment, we are able to capture 300 cells. So we can see different stage. We are able to identify different stage of oocytes. And each cells actually have more than 10,000 gene can be detected because the size is very big. The gene capture efficiency on our platforms is related to the size of the surface. because they're a very large surface. So we are able to capture more genes than standard cells. That's why we can able to reach to more than like 10,000 cells or 10,000 genes per cell. So, and interesting thing is we also observed that different genes in different cells have different subcellular kind of like profilings. So, for example, some like genes are only showing in the are random in oocytes, but some are only like distributed in the central. So maybe because they play different functions. It's because this is very early stage. We didn't go in detail to do the analysis. Later, we're now using a more kind of like a high resolution imaging technology combined with expression patterns to see if we can identify certain kind of like interesting patterns of genes in oocytes. Actually, before I showing you that we can using the 6 centimeters chips for milling cells, but theoretically in like 1 centimeter chips, we can put 1 million cells in 1 centimeters. Before we didn't do is because we are like kind of like want to kind of like reduce the risk of like a cell overlays with the other cells. But actually, you can directly put 1 million cells in 1 chips. So recently, we able to carry out this different experimental, able to capture like almost like 1 million cells in 1 centimeter chips. And when we sequencing like either like a low kind of like a low number of reads or like a high number of reads, for example, 34 giga reads or 3 giga reads, we can still have like a very good numbers of genes per cells. For example, in this, even in this like 3 giga reads, we can still have like 165 genes per cell, and in for like 1 million cells. So it's quite interesting because this could be a very like low cost, like a high throughput, a million cell level single cell analysis. And we further trying to reduce the sequencing cost by target gene capture, because for certain type of cells, so because we already know they're like marker genes, so we can only focus on those like target genes. We don't have to sequencing very deep on their like housekeeping genes. So here we're showing that we do using this like a founded design this like a 500 target genes. Like each genes can able to enrich like around 550 fold enrichment, but only with like 1% of sequencing data. So which means we can using this like a very low cost, like just 1 centimeter chips, but only like less than 1 giga reads to generate a million cell single cell sequencing data. So it's kind of like a huge data set for a lot of like downstream analysis. So it's quite interesting. This is not published in this science paper, but it could be a very interesting like further expansions from current technology. And because of that, then we can think about, can this technology to replace the current flow cytometry analysis for the clinical diagnosis applications. So we actually try a little bit, and seems like for different sample type from the blood, from the urine, from different kind of like fluidic like clinical samples, we can actually capture those like disease cells and able to identify those like for example the cancer like cells in these urine samples. So it's quite interesting. And we also trying to capture the circulating tumor cells from human blood. Before we using like scientists using different kind of like enriched methods to capture what they call the CTC, but it's not very successful. It's not very popular right now. But in our platforms, we actually can capture those like circulating cancer cells in our platforms, and we can also see like different cells attached together in human blood for the cancer patient. It's probably could be used for this like a cancer early diagnosis, but this is still in the early stage. So this is actually a kind of like summary of these platforms. So it's kind of like a scalable and unbiased cell capture platforms, and it can integrate it with different multiomics, including the cell like imaging data, and it's also can capture the cells in the culture systems in situ culture, and can detect the cell-cell interactions and the extracellular vesicles. And it can also be used to the analysis for like very large cells for the subcellular structures for those large cells. So this is the information I want to share today. So, thank you.
Wonderful. Let's take a moment to thank our speaker for the absolutely wonderful presentation. I gotta say, it was quite something to look at. The visuals were stunning throughout, very engaging. So with that, I'd like to open the floor for any additional questions that the audience may have. And actually, I would like to start off with a question myself. Mm-hmm. So this method is amazing, right? It has all these different sources of information and many particularities that are, well, of course, method specific. From the modeling side of things, how would you integrate all these different sources of information in one comprehensive manner so as to actually make the modeling choices successfully integrate these paired modes of information within the data?
So your question is how we can integrate different layers of information together, right?
That's exactly right.
Yeah, so because we have the barcodes of all these like molecular informations, and this barcode, the barcode is also able to have their physical locations on the chips. As I showing you earlier, that because on our chips, we actually have like each dot has a unique barcode, and they have like physical locations after sequencing. So it's very precise locations. So this is kind of like a coordinations that you can use to integrate different informations. For example, you're sequencing the RNA has this barcode. You capture the imaging of the cells. The imaging, they have the physical locations. They actually are integrated together by these physical locations.
I see. Thank you. Do we have any more questions from the audience?
Yeah, actually, I have a bunch of questions. So the first question is like, from there is a page to say there are many sequencing techniques, and stereo cell is like actually the more advanced nowadays to be both multimodal and also across like hundreds of millions of cells, maybe 1-2 slides before. So I wonder, like, because a lot of people would know about this 10x technology because they have published their data in this in this database called cell by gene database. So I'm wondering, like, whether stereo cell would also consider similar things to release a large data set for like global scientists to work on it.
Yes, I saw the CZI is initiate this like 1 billion cells projects, and those HCA projects actually already have a very large data set right now. So right now we are working on similar kind of projects aiming from like 1 billion to 10 billion cells. So we actually have also have a database. If you search in the db.cngb.org, you will find the database.
Okay, CNDB.org.
I can put it here if you
Okay,
can.
thanks.
Yeah, you can see it.
Oh, DBCNGB. Oh, thank you.
Yeah.
Thank you for sharing. And also, I have some other questions. It's more research oriented. So actually, I'm very interested in SciSig because I believe that actually when we're sequencing the proteins, we are like further closer to the disease directly, especially for the cell immunology. So I'm wondering, like, is there any like further cool things you can think of, like we can use SciSig to do, like, for example, because I'm a researcher on the machine learning side, but I'm trying to use like deep learning to tackle these biological problems. So I'm wondering what kind of problems could be important and also have large impact when we try to leverage the SciSig data.
Yeah. So sometimes, when we are just using like one layers of information for the only transcripts, like trying to build up like what they call the cell language model, those kind of things, it could be difficult because it's the only RNA information is not giving you all these details.
Yes.
So that's why not only the protein informations, but also the imaging informations. If you are able to see the real cells, what it looks like, you can already, because transcript data is kind of like has a lot of like missing informations. Like the current technology, all this technology cannot sequencing capture all of them. Probably like 80% of the RNA information is missing from the current data. But the protein data as well as the imaging data is much higher dense compared to RNA sequencing. So although RNA sequencing is much higher throughput, but I still believe that it should be combined with the imaging data as well as the protein data.
May I ask, like, how many proteins can stereo cell SciSig sequence? Because like I know that so before, like for example the CyTOF, they can only sequence like 40 to 50 proteins per cell, and also for other like more high throughput sequencing techniques for proteins, they might do like 200s or even 2,000s of proteins, but they are quite low throughput.
So it's really limited by the number of antibody you have. So you can, because the conjugation methods is very simple. So as long as you have like high numbers of antibodies, you can conjugate your own target panels. So our like maximal like experience, we have more than like 500 antibody targets, at the same time. So there's no limitations on it. So if you have like 1,000 of different antibodies, you can like label them separately and working like sequence them like just in once.
Okay, thanks. Yeah, that's very cool. Also, sorry, like one last question. Because you also mentioned like the cell language model. So I believe that nowadays like the virtual cell idea is quite popular, where people are trying to do perturbation data. So I think like from your introduction for the stereo cell, because you can do cell culture to see how cells are actually evolving and interacting with each other. I think it should be possible for stereo cell to also do perturbation data, right?
Yes. So that's why I introduced like a milling cell in one S1 chips. So it's kind of like a concept if you do random perturbations, so you have to sequencing a lot of them, able to find out that. So the idea is kind of like in one chips, you have the whole a million cells which can capture majority of those perturbation results and have a lot of redundant data to capture to confirm the results. So and you have and if you have the antibody as well as the imaging data, you probably can see how this a perturbation change the function of the cells.
Yeah, okay. Oh, that's very, very cool. Thank you. Yeah, this technology is really amazing. Thank you so much. Maybe any other questions from the audience?
Yes, hello again.
Yeah.
I have two questions. First, I probably misunderstood. How can you confirm that you can at least preserve the real spatial distribution of the cells? It's not just spatially enhanced so that you can visualize each cell on the plate. It's like, can you confirm that you can really preserve the real spatiality of your original sample?
Yes. So this is probably a better slide to explain your questions. So as you can see that this is trying to see if we can detect cell-cell interactions.
Mm-hmm.
So to detect that, we have to preserve the real locations of the cells.
Yes.
Yeah. So in the imaging data, this imaging data is the same data of the transcript data.
Okay, so it's not just cells that you mix together and then you put them randomly on a plate and you visually observe them. Okay.
Yes.
Okay. Second question, pretty simple. What do you think are the limits of your technology today?
The current limits, one of the major limits is the number of the transcripts is still not high enough. So currently, our technology, as the same as other commercial high throughput technologies, can only capture few thousand. But ideally, if we can capture, for example, 10,000, then we have, especially for those low expressed genes, probably those low expressed transcript factors probably much more important than this high express level housekeeping genes. So it's kind of a limitations. So the second limitations is that currently we still have the data usage efficiency is still low compared to the other fluidic-based technology, because we in the chips we not only capture the RNA from the cells, but we also have a lot of backgrounds, which is kind of a lot of waste data set. That's why we right now working on the target enrichment to increase data usage efficiency.
Great, thank you.
Do we have any more questions from the audience? Yeah, Dylan, please.
Yeah, hi, can you hear me?
Yes.
So I was kind of blown away by that slide that you had about an older piece of work you had about the 3D macaque spatial reconstruction. Yeah, that exactly, the middle column there. And of course, what this has potential to do also is to enable alignment with other brain imaging modalities, right? So if we go back to the discussions we've been having about stereo cell site, I wonder if stereo cell site could be used to kind of expose the distribution of different surface cell receptors to then know which kind of radio tracers you might be using for PET imaging in a way that then once you have the PET images, you could then align them to like these 3D atlases and figure out what are like the different biomolecular activities based on brain imaging, conjoined with the actual transcript levels that your technology is profiling. So basically my question about kind of multimodal integration between something like stereo cell site and brain imaging.
Mm-hmm. Yeah, this is a very interesting questions, and because we're also working with a lot of like neuron scientists, so they are also kind of like trained to do that. Currently, they are using the other methods to do the 3D, like what they do, the connect points, trying to see how different neuron cells are connected to each other. The current problems of doing that is because we have to do the tissue slides. And it's really difficult to do the tissue do the alignment between two adjacent slides together to just tracing one molecular. The current like a 3D reconstructions using the current stereo-seq and the stereo-site data is still like not able to do the alignment cell to cell. It's more like alignment to the region to region. So that's kind of like a current bottleneck. If we want, but the other technology, like but low throughput one, like FMOST, for example, the FMOST technology is able to do the alignment to the cell to cells, but they can only tracing like one antibody, for example. So they already have such kind of technology, but they cannot do sequencing because they have to cut out one layers of cells and to see this like images from the block surface of the tissues and cut another layers. So, but they are not able to cut the tissue slides and to do the analysis on that slides. Yeah, the simple answer to that question is we tried, but we didn't able to do that.
Yeah, it's a hard challenge. Thanks for the answer.
Do we have any more questions? If not, well, let's thank our speaker once more. Thank you very much for coming out, and that concludes the recording of the 30th edition of the Multiomics Reading Group. Thank you, everybody.